Okay, so you can see here that we have the phospholipid bilayer and we have some proteins, and this will make up the plasma cell membrane. And I've split it into 3 sections, and all of these are colored very specifically. So if you look here, the one here that is red, orange, and red, and yes, that is orange in the middle, it doesn't show up too well, that would be carbon dioxide. Okay. Here, the one that's just 2 red ones, that's oxygen. Over here, the red and the 2 little ones, that will be 2 hydrogens and 1 oxygen, H2O, so that would be water, H2O. And then over here, the green triangles represent sodium ions, and the pink circles represent, the green triangles are potassium ions, and the pink circles are sodium ions. And so, yes, they're all colored, and I've separated it because all these things use different techniques to get in and out of the cell. So here's the cell membrane. On the top, this is intracellular, so this is inside the cell, and this is extracellular, so this is outside the cell. So you could literally put it like that, would probably make it a little easier to understand. Okay, so here's the cell membrane, inside the cell, outside the cell. Okay, so the key here is different materials have different properties, and the different properties determine partly how they go in and out. And I'm going to move this because it's easier to record it this way. So I always have this little technique here. So I'm going to look here and I'm going to count, right? I need to know where my concentration gradients are. And so if you look here, I got 1, 2, 3, 4, 5, 6, 7. I've got 7 lots of oxygen on this side of the cell membrane. And over here, I got 1, 2, 3. So I've got 3 lots here. Okay. If you remember from our concentration gradient, okay, things like to go with the concentration gradient. We're going from where there's more of it to where there's less of it. Okay. So my oxygen, I've got more of it here and less of it here. So my oxygen is going to go from this side of the cell membrane into the cell where there's less of it. Okay, we're going to go from high to low. Right? Now, what do we call the spreading out of materials from where you've got more of it to where you've got less of it? We call that diffusion. And so oxygen is going to pass directly through the cell membrane, phospholipid bilayer, by simple diffusion. Okay, that's what I've written down here, simple diffusion. Okay, now, and then here's a real good clue. If you get a diagram that looks like this, look at the direction of the arrow. So if the arrow is going this way, I know that this material here is going from here to here. Because I'm going from 7 to 3 molecules, I'm going from a high number to a low number. I'm going from high number to a low number. I'm going with the concentration gradient, and so this is going to be passive because I'm going downhill from high to low. Okay, now carbon dioxide, if I look at carbon dioxide, I've got 1, 2 here, right? And I've got 1, 2, 3, 4, 5, 6, 7, 8 over here, right? And I'm going to draw an arrow, and the arrow is going to be pointing like that, which means that carbon dioxide is going to go from here, where there's more of it, to where there's less of it. Okay? Concentration, even though it's going in the opposite direction to the oxygen, it's still going from where there's more of it to where there's less of it. It's going from 8 to 2. So it's still going from 8 to 2. It's still going from high to low. So carbon dioxide is also passing through the cell membrane by simple diffusion. And because it's going from where there's more of it to where there's less of it. It is also passive. It doesn't require energy. Okay? Now, these two things here are what we call non-oxygen and carbon dioxide. They are what we call non-polar molecules. Right? They're small, they're simple, and they are not polar. That means that they don't have any form of electrical charge, so it's real easy for them to go straight through the cell membrane. Okay? And that's what things which pass straight through this cell membrane, we call this simple diffusion. Diffusion is where things spread out from where there's more of it to where there's less of it. Now, let's look at here. We got water molecules. And the water molecules, if you remember, when you look at the cell membrane, that phospholipid bilayer, the heads are hydrophilic. That means they love water. But the tails are hydrophobic. It means they hate or repel water. So water molecules, they can't just go straight through the phospholipid bilayer because they're okay once they get here, but they can't get through all the tails because the tails hate water. So in order to help that, what we have here is a channel protein. Okay? It is a protein that has, right, a kind of tube that runs down the middle. And this channel protein is a very specialized type. It's called an aquaporin. Okay? Now, if you ever see aqua, you know it's got something to do with water. And a pore is like, like you have sweat pores and things that, those tiny little holes in your skin that get clogged up with stuff. Okay? An aquaporin is a tiny tube. So this type of protein molecule here is an aquaporin. And its job is to help water molecules pass through the cell membrane. And again, right, I'm going to put my arrow in this direction. So let's look. 1, 2, 3, 4, 5, 6. Is that 6? 1, 2, 3, 4, 5. No, we've got 7 water molecules on this side, and we've got 3 water molecules on this side. So my water molecules are traveling from high, where there's more of it, to where there's less of it. So they are, again, going from where there's a high amount of water to where there's a low amount of water. They're going with the concentration gradient. And they're being helped out by this protein molecule that's got this tube that goes through the middle. This is also a type of diffusion, but because the protein molecule here is helping it, right? When you help someone, you facilitate, right? And so this is called facilitated diffusion. It's still passive because we're going from where there's more to where there's less, right? But we're using this channel protein, specifically this one is an aquaporin, we're using it to pass through the cell membrane. So, how does water get in and out of the cell? It gets in and out of the cell by facilitated diffusion. Okay? Remember, it needs to have the protein to help it through, because otherwise it would get stuck and repelled by those hydrophobic tails of the phospholipid. Okay, the next one is this, right? And I've already drawn the arrows on this one. This is an example of what's known as a pump. And a huge clue, if you see ATP, you know that this is active. That if it's active, you know we're going from where there's less of something, we're trying to put it into somewhere where they've already got more of it. So if you look here, you will see that there is less potassium on the outside and more potassium here, right? So the potassium, they sit in here, right? You come in here, one, and you would have another one fit in there. Then we expend some ATP, and it changes shape. So we start like this, we get 2 come in here, and it changes shape, and then it gets dumped on the other side. So you can see that sodium here is going from a low concentration on the outside to a high concentration on the inside. So it's going against the concentration gradient. It's going from low to high. But at the same time, potassium ions, right, these little ones here, they're loading up on this side, and when it changes shape, they get dumped out on the other side. Okay, so what we've got here is less sodium is moving to where there's more sodium, and less potassium is moving to where there's more potassium. And this is an example, this is the most famous example of active transport. It is called the sodium potassium pump. Okay, because we are constantly pumping sodium out of our cells and pumping potassium into our cells, and they are both going against their concentration gradients. So you see the ATP here means it's active. So these two here are an example of active transport. Okay, active transport, because we are putting stuff in and out of the cells against the concentration gradient. The key thing is, how do I know it's against concentration gradient? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. There's 11 sodium here. There's 3 sodium here. And we're going from 3, right, to 11. So we're going uphill. If you count the potassium, 1, 2, 3, 4 on this side, and notice I write the numbers down. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 on this side. And the sodium's going from where there's 4 to where there's 12. From a low number to a high number. So again, it's also going against the concentration gradient. So if you get a test question that shows you something, it might not show you all of this. It will only show you like one bit. Step 1, count the molecules on each side of the arrow, of the cell membrane. Right? Write those numbers down. Look at the direction of the arrows. Are we going from a big number to a small number, or are we going from a small number to a big number? Right? Which is it? If it's if it's a big number to a small number, we're going big to small. We're going with the concentration gradient. If it's a small number to a big number, we're going against the concentration gradient. We're going uphill. Big number to small number, passive, doesn't require energy. Small number to big number, active, it does require energy, and there's usually a clue here. So count the numbers, look at the direction of the arrows, work out is it active or passive. Then confirm if it's active, you'll see ATP, and usually the question is, how does oxygen get into the cell membrane? It goes through the phospholipid bilayer by simple diffusion, because it goes from a high concentration to a low concentration. How does sodium get into or out of the cell? It's going against the concentration gradient using a pump, which is active. So it's using energy. Okay, I hope that helps. Add these labels to your diagram. There is a picture of this in along with the rest of the assignment.